Hi everyone, I am Mr.B.Suriya Sabarinath, working as an assistant professor in the Faculty of Agricultural Sciences from Bharat Institute of Higher Education and Research. I am handling the course of Agricultural Microbiology. So in this video, we are going to discuss about what is meant by mycorrhizae and what is meant by vesicular and vesicular mycorrhizae and its mass production and isolation and morphological observation. So before going to the video, so first we are know about the importance of mycorrhizae. So mycorrhizae means myco means fungi, rhizae means root. Fungus will be associated with the plant root, so that is called mycorrhizae. So in this association, the plant will be protected by fungi. It will be protected some root knot nematodes and also some of the harmful root-borne infection and the soil-borne infection from the pathogens, and also it will mobilize the phosphorus. So now what is mean by mycorrhizae? So the term mycorrhizae was taken from Greek language, that meaning fungus root. So it was coined by Frank in the year of 1885. So we already studied this. Frank is the scientist, will be coined the term actinomyces symbiosis, and also he isolated the microorganisms is called Frankia. Then the mycorrhizae is the mutual association between fungal mycelia and plant roots. So Vamp, vesicular mycorrhizae is an endotrophic, that means live inside of the plant. So this mycorrhizae formed by asymptoted phycomycetes fungi. Vamp helps in nutrient transfer mainly of phosphorus, zinc and sulfur. So this is the picture. So it is the Vamp. So you see the mycorrhizae. Mycorrhizae means we already discussed it. The fungal will be associated with the plant roots. So you see that now you see the hyphae of the plant root, the fungal hyphae that is present into the plant root. So groups of mycorrhizae. So there are several groups. So the first group is Ectomycorrhizae. So Ectomycorrhizae means association or comes under Basidiomycotina and Ascomycotina. So example is Amanita, Suillus, Laccaria laccata. Endomycorrhizae. It is the next association. So the Endomycorrhizae. So both Ectomycorrhizae and Endomycorrhizae. So these Ectomycorrhizae means fungus will be present outside of the plant. Endomycorrhizae means present inside of the plant. So these endomycorrhizae, they have further classification. First one is arbatoid mycorrhizae, association with some species of ericaceous plants. So example is Basidiomycotina and Ascomycotina. So Basidiomycotina is also called as club fungi. Okay, Ascomycotina will produce the ascus. Next class of endomycorrhizae, that is orchid mycorrhizae. It is associated with the orchid plants. Example is Rhizoctonia and Armillaria. Next classification, that is arbosicular mycorrhizae. So out of this mycorrhizae, arbosicular mycorrhizae is associated with most of the plant families. So it will supply the nutrient as well as it gives the protection to the plant against harmful pathogen. Ectomycorrhizae. So now we see the definition of ectomycorrhizae. So that means lives only outside of the root. It forms mainly with the conifers and oaks. So this is the picture of ectomycorrhizae. So this picture is the structure of ectomycorrhizae. You see the xylem, epidermis, or cortex, and the harpic net and the fungal sheath. Okay, this is the structure of root. In this root, it will associated with some fungus. Okay, so what is meant by harpic net? So harpic net is nothing but it is the hyphae that fungal penetrate the root cortex tissue and acts as a storage and transport organ of phosphorus. So the benefits of harpic net. So it is protection from pathogen. It produced endospores. So endospores are reproductive structure of fungi. The next one is it produced polysaccharides that is called glomalin. Next classification, that is Endomycorrhizae. Endomycorrhizae means it present inside the fungus plant, inside the fungus in plants. Exchange mechanism and the inside of the root and the hyphae extend outside the root. Endomycorrhizae form mostly with green leafy plants and most commercially produced plants. Example, most vegetables, grasses, flowers, fruit trees, and ornamentals. So this is the structure of Endomycorrhizae. So in this Endomycorrhizae, the Chlamydospores was found. So here there is no hard pigment is found because here the vesicles and the arbesicles are present. So both will be act as a storage and transporting organs of the plant. Okay, so epidermis layer that is used to protect the root. So in that bottom, the endodermis layer are present. Okay, so the root hairs. Okay, the root, this is the root hair. The size of the size is around 100 micrometer. Okay. So the difference between ecto and endomycorrhizae, you see the what is it called? That hyphae. Okay, here the hyphae do not penetrate the root cells. Okay, but here the hyphae will penetrate in the root cells and it produces the vesicles and the arbesicles. Ericoid mycorrhizae. It is also the class of Endomycorrhizae. It was found in inhospitable environments, particularly acidic environment. The fungi involved in the symbiotic relationships are Ascomycota. Despite the harsh conditions, the mycorrhizae are still able to take up nitrogen and the phosphorus of the host plant. It also helps to acquisition of iron, manganese, and aluminum ions. So these iron and aluminum ions will be fixed the phosphorus in various soil. So far, if the pH is become acidic, means the phosphorus will be fixed as aluminum phosphate as well as iron phosphate, or sometimes it is fixed as calcium phosphate. So which are often present in highly available forms in acidic soil. Next one, orchid mycorrhizae. So at some point of their life cycle, orchids through a period of time where they are not photosynthetic. Photosynthetic means it cannot do photosynthesis. That means the plant, the lack of chlorophyll. So so that only the photosynthetic will not be happen. So during the time, orchid cannot perform photosynthesis or manufactures its own carbohydrates. So it must rely upon mycorrhizal fungi to provide it with nutrients. So in this association, if there is no mycorrhizae, means the plant will be died because it will supply the carbohydrates to the plants. So generally, orchid is non-photosynthetic when the orchid is the seedling stage of its life. Next one is arbutoid mycorrhizae. Arbutoid mycorrhizae are found in plant genera Archastophilus and Arbutus. The fungi that from arbutoid relationships are Basidiomycetes. So that is called club fungi. Okay, mushrooms also comes under the class of Basidiomycetes. So it associates fungal sheath or mantle covers the root of the host plant, similar to the structure of ectomycorrhizal associations. Next one is monotropid mycorrhizae. So monotropid mycorrhizae throughout to be part of the arbutoid mycorrhizae group. While arbutoid mycorrhizae hyphae penetrate and form extensive structures within the cells of host plant. Monotropid mycorrhizae do not penetrate the cell walls of the host plant. They don't produce the chlorophyll. So if there is they don't produce the chlorophyll, means it is also non-photosynthetic. It get carbohydrates from and other nutrients from mycorrhizae. Next one is Vamp, vesicular arbesicular mycorrhizae. Now it is called as arbesicular mycorrhizal fungi. So the Vamp fungi are symbiotic, that means mutually benefited endotrophic present inside mycorrhizal fungi found in the root of higher plants. They are included the family Endogonaceae of Zycomycetes. Vamp fungus infects a plant root and forms vesicles and arbuscules in the root cortex and a permanent mantle of hyphae on the root surface. The vesicles are large bulb-like structure used for storage of food materials. Then arbuscules are modified from hyphae as coiled or densely branched finger-like structure like Astoria, and they are involved in interchange of materials like nutrients between the plants and the fungus. So the vesicles that is used to store the food material means nutrients. Okay, arbuscules that will use helps to transport the nutrients. So the vamp found in many crops like rice, maize, potato, soybean, cotton, tobacco, sugarcane, tomato, rubber, strawberry, coffee, tea, cocoa, peas, apples, and papaya. So this is the structure of worm. So the worm will be classified as several species. So before that, how it is be classified means based on this chlamydospores only. Okay, so this one is chlamydospores. Okay, it's in this external mycelium. This layer is epidermal layer, and this one is Casparian strip and endodermis layer. This one is vesicles, that is a bulb-like structure used to storage of nutrients, and this one is arbuscules that is used to transport the nutrients. Okay, so based on that chlamydospores, and this one is stalk. Okay, so the stalk wise the classification will be divided. Okay, now we discuss about the classification. Okay, so the genera of Vamp fungi. So Gredman and Trapp having recognized the genera of Vamp fungi based on the spore morphology in the year of 1973. So the characters are, first one is Glomus. So Glomus means chlamydospores with a single stalk. Okay, in this previous picture, I showed the chlamydospores and the stalk. If it is a single stalk, means that is classified as Glomus. Example is Glomus microcarpus, Glomus mosaic, etc. Next one is Gigaspora. So chlamydospores with a bulbous stalk. So here the stalks are bulbous. Okay, the example is Gigaspora nigra. Third one is Aqualaspora. So chlamydospores are sessile. So the example is Aqualaspora scrobiticula. Next one is Sclerocyst. Chlamydospores are thick-walled. Example is Sclerocyst clavispora. And last class, that is endogene zygospores inside the sporocarp. So out of this 5 classification, endogene is somewhat different compared to the Glomus, Gigaspora, Aqualaspora, and Sclerocyst. Okay, the species of endogene that is called endogene incresta. Morphology of Vamp. So Vamp has 3 distinct regions. First one is external hyphae, second one is arbuscules, third one is vesicles. External hyphae are aseptate. So what is meant by aseptate? So there is no cross wall. Septate means it having cross walls. Okay, dimorphic. Okay, different morphological characters and thick-walled and closely oppressed on the root surface. At the point of contact to the plant root, it bears an approsoria. An arbuscule is a rectomosely branched, brush-like haustorium produced at the tip of fungal hyphae in a cortical cell. It gets digested as the host cell matures. The vesicles is a spherical or oval, thick-walled structure produced at the tip of hyphae in the intracellular space. It is rich in fat droplets and hence serve as storage organs. So the spores germinate into a hyphae called permanent hyphae on the root surface. This hyphae is aseptate and thick-walled. Aseptate means it doesn't have septa, so that means there is no cross walls. Okay. Thin-walled, short-lived hyphae arise from the permanent hyphae and penetrate root hairs or epidermis to reach the cortex. In the cortex, they grow through the intercellular spaces between the cells. Tips of these hyphae enter the cells and form arbuscules and vesicles. Some other thin-walled hyphae come out of the plant root and produce spores that is called clavidospores. Then now we discuss about importance of Vamp fungi. Vamp fungi helps to plants to intake more amount of zinc, sulfur, copper, phosphorus, potash, iron, manganese, and boron from the soil. Vamp fungal infections increase the growth rate in plants. Example, citrus, maize, wheat, barley, etc. Vamp fungal infections increase the absorption of water by plants from the soil while drought prevails. Vamp fungal infections increase the concentration of cytokinins and chlorophylls in the plants. It reduces the sensitivity of crops towards high level of salts and heavy metals in the soil. So it improves the hardiness of transplant stalks by serving as extra root hairs. Example, pine. Vamp provides resistance to plants against various soil-borne plant pathogens causing root diseases. In fumigated soil, plants show stunted growth. Vamp fungal infection reduce the stunting of plants in such soils. Vamp fungal infection increase the yield in crops like potato, maize, barley, etc. When the infected plant is starving for food, Vamp gives the plant its own food and protect the plant. So this research was done by Heyman in the year of 1974. So now we are discussed what is meant by Mycorrhizae, what are the classes of Mycorrhizae, and what is meant by vesicular or vesicular Mycorrhizae and its morphology. So now we are going to discuss about isolation of vesicular or vesicular Mycorrhizae. For isolation, there are two types. First one is direct examination of infected root. Another one is examination of Vamp from soil. Okay, the spore counting method. Okay, that was done by wet sieving and decanting method. So first we discuss about examination of Vamp infection in root. So first, what are the steps involved means? First step is root clearing and staining technique. First, you should collect the root. Okay, so collection of representative root samples from growing roots of plants. So preferably thin roots and wash gently in running tap water to remove adhering soil particles. Next one, clearing of roots. So cut the root into 1 centimeter pieces and transfer to boiling tubes. So add 10% of KOH solution till the root immersed. Plug the tubes with cotton and cover with the paper. Heat at 90 degrees Celsius for about 1 hour. Alternately, cook the samples in autoclave at 100 to 120 degrees Celsius for 15 minutes. Care should be taken to avoid smashing of root. So delicate roots can be heated for a short time with care. This boiling with KOH leads to clearing of root content. Wash the root till no brown color disappear in washed water. Okay, if the brown color will not be disappear means you will be do bleaching, or else you directly go for the next step, acidification. The next one is bleaching. So if Bleaching is necessary with the heavy pigmented root material which is not clearly adequately in KOH alone. Okay. So immerse the root in alkaline H2O2 solution at room temperature for 10 minutes to 1 hour. So till the roots are bleached, alkaline H2O2 is prepared by adding 3 ml of NH4OH, 30 ml of 10% H2O2 to 567 ml of tap water. So wash the roots thoroughly in water to remove all H2O2. Okay, so after bleaching is done, we should go for acidification. So if the root is not clear until the brown color is not disappeared, means then only you go for bleaching. Okay, or else you directly come to this acidification. Acidification is done by immerse the roots with 2% hydrochloric acid and retain it for 3 to 5 minutes and decant. After acidification, should not wash the roots in water since the stains requires an acidic medium for the reaction. After acidification, we should do the staining. So if the stain, due to the excess stain in the slide, then only you should do destaining. Destaining is not necessary. Bleaching is not necessary. If KOH is not disappear means you should do bleaching, and also destaining will be used if you should apply more amount of stains means we will do destaining. Okay, first we have discussed about staining. So add equal amount of 0.05% trypan blue stain. So trypan blue stain is only used for Vamp for identification. So 0.05% of trypan blue stain in autoclave for 10 minutes at 120 degrees Celsius or heat at 90 degrees Celsius for 15 to 20 minutes or left unheated for several hours. If roots are stained excess, immerse the root in lactophenol for destaining. So after this root examination, you should collect the root and you should be arrange root segments in lengthwise on a slide with fine forceps. So and next by using cover slip and mounted. So add a small drop of PVLG. So that means polyvinyl lactic acid glycerol. Mounted at one end, then slowly lower cover slip at that east end first. Okay, so allow mounted to flow around roots before gently tapping cover slips to flatten roots and remove air bubbles. Okay, so this is the steps by using that mounting. So please do it in your lab. So assessing Mycorrhizae mounted on slides. So randomly selected microscope field and view and field of view and cross hair position. So this is the microscopic view of that mounted slide. So first you should see the hyphae and the vesicles. Second, you have to see hyphae only. Third one hyphae and arbuscules. And finally you will see the root. Okay. So these are the possible intersects you should be visible on the microscope. So this is the one more method for isolating the Vamp. So first you should be sample collected from the pot culture with the soil cover, then wash the root from the spore, and you should be examine the root in the petri plate, and you should be quantify and identify the Vamp spores with the help of that stereozoom microscope. Stereozoom microscope only you should be examine the Vamp roots, then clear and stain the roots. So here you should be stain the roots with the trypan blue stain. Okay, then use visual examination and guide line in intersect method to quantifying of Vamp. Okay, guide line means you should be draw the grids. Okay, then make slides from root and identify fungi using morphology. Okay, you have to after you should be visual estimation of Vamp. You should do the mounting and you should be preserve the slide. The next one is recovery of Am fungus force from soil by wet sieving and decanting technique. So this is the another method. So in this wet sieving and decanting method was discovered by Gradman and Nicholson. So for this method, first you have to collect 100 gram of soil in 1 liter beaker after removing stones and other bigger sized debris, etc. Pour 1 liter of water and stir well with a glass rod so as to detach spores from soil particles. Allow the soil particles to settle down for few seconds. Transfer the water to the sieves arranged in the descending order. So here you have to take the sieves by descending order. So the sieve size it will be 45 microns, 90 microns, 180 microns, and 725 microns. Again, pour the water in the beaker and stir well. Allow to settle for seconds and transfer into sieve arrangements. Repeat the process 4 to 5 times till the suspension is become clear. So collect the sieving from each sieve using fine get of water through sequence bottle into a small beaker. Okay. For taking total count, pass the sieving through the fine sieve, 45 microns. So here you take 4 sieves. Out of that 4 sieves, we will take 45 micron sieve. So that is the lowest micron. So what is the observation means? So that it will be eliminate some of the microns from sizes enlarged above 45 microns. Okay, so collect the sieves again in a beaker from the fine sieve and make up to 10 ml. So transfer the known volume 1 ml of the sieving into a gridded Petri plate and observe the plate under the stereozoom microscope. So all this experiment I will be displayed in that previous picture. Okay. So now we are scan the grades under one by one and look of the spores. So count the number of spores present in the Petri plate. So here the observation is the chlamydospores will look bigger and thick walled. Okay. If it is crushed with the needle means the fatty substances will be oozed out from the spore. So this is the microscopic view. So especially in stereozoom microscopic view, you see the chlamydospores. Okay, it should be oozes out at the time you will be touched with the needle. It should be oozes out. So now we have to study about Vamp and what is the isolation procedure. Okay, so this is very useful to preparing the mother culture. Okay, right. So now we are going to discuss about Vamp fungi mass production. Mass production means we will produce a much more quantity. So that is called mass production. So this Vamp mass production will not done under any laboratory. Okay. So arbosicular mycorrhiza cannot be produced in laboratory using any artificial media. So because the root is the medium for arbosicular mycorrhiza. So it is an obligate symbiont in nature. It can colonize maize and grass. So maize plant is used as a host plant for mass production of Am fungi. So vermiculite is the best carrier material for production of Am fungi. So now we discussed about mother culture. So mother culture can be produced from efficient spores using funnel techniques as well as multiplication in small sized pots. 3 to 4 cycles will be made for developing the mother culture. So then only it's become very virulent and also it's effective. Okay, the number of colonization percentage will be we get more. Okay, so the mother culture should have 100% root colonization and the minimum of 8 to 10 spores per gram of the inoculums. Mother culture can also be maintained in maize roots continuously. So method of production of. So first you will be form a trench in the field. So the size of the trench is 3 meter, 1 meter and 0.3 meter length, breadth and width and line with the black polythene sheet. So this sheet is used for plant growth tub. So after harvesting, you should take to pull out the tub and you will be collect the roots. Okay. So the root is the economic part for the mass production of Vamp. So mix 500 kg of vermiculite and 50 kg of sterilized soil. So before doing mass production, you should be sterilized and packed in the trench up to height of 20 centimeter. Okay, height should be 20 centimeter. Spread 10 kg of AM inoculum mother culture. Okay, so there is a mother culture at 2 to 5 centimeter below of the surface of the vermiculite. So now you raise it the bed up to 20 centimeter, but you should inoculate in between 2 to 5 centimeter. So surface sterilized maize seeds. So the sterilized will be seeds will be sterilized with the sodium hypochlorite for 2 minutes. At the time of sowing, you should apply 20 gram of urea, 20 gram of single superphosphate, then 10 gram of potash. So you should apply muriate of potash. Okay. So regularly water the plants, grow the plants for 60 days. For commercially, the maize plants will be grow around 90 to 120 days. For cultivation of Vamp, 60 days is enough because we have cultivated only the roots. Okay. After 60th day, cut the root and mix it with the vermiculite in the trench. So inoculum is produced to consist of vermiculite, spores, pieces of hyphae, and infected roots. So within 60 days, 55 kg of AM inoculum could be prepared from 1 square meter area. So for 1 cent, that is around 40 meters square. Okay. So this inoculum will be sufficient to treat 550 meters square nursery or area having 11,000 of seedlings. Okay. So now you have to see. So this is the mass production. Okay, under a Vamp inside in a brick-lined tank and the mass production of Vamp inside a cement tank. So here you should use the okay, the polythene cover that serves as a tub. Okay, you should cut the root and you should mix it with that vermiculite. Then you will be do the packing. So for that packing, you should follow the quality standards such as name of the biofertilizer and what it should be recommended for, the name of the manufacturer, for which crop it is recommended, and what is the quantity, date of packing, date of expiry. So all the details you should be mentioned in that. Okay. So field application. So after mass production, so what are the method of field application? So Vamp fungal inoculant is diluted with water and mixed with the seeds to make a pellet of inoculant on them. The seeds are then sown in a main field as usual. In another method, the inoculant is spread uniformly all over the field before plowing, and the crops are transplanted of seeds are shown in the field as usual. Okay. So first one, nursery application. So nursery application, in the nursery application, 100 gram of inoculum is sufficient for 1 meter square. The inoculum should be applied at 2 to 3 centimeter below the soil at the time of sowing. Okay, the seeds cutting should be shown planted above the AM inoculum. Next method is for polythene bag raised seedling. So it is especially for forest tree species, coffee and tree. So that is a plantation crops. So about 10 gram of inoculum is sufficient for each plant raised in poly bags. Mix 10 kg of inoculum within 1000 kg of potting mixture and pack the potting mixture in polythene bags before sowing. For outplanting, that means transplanting, 20 gram of AM inoculum is required per seedling. Apply inoculum at the time of planting. Okay. So per seedling means per kg of seedling. Okay. For existing trees, 50 to 100 gram of AM inoculum is required for inoculating one tree. Okay. So apply inoculant near the root surface at the time of fertilizer application. So that only the root will easily absorb the Vamp spores and it should be in a beneficial way. That means mutually benefited. So the plant is a shelter for Vamp. Vamp will provide nutrients to the plant for their growth and development. So I think in this class we are discussing about mycorrhizae and types and what is meant by vesicular, vesicular mycorrhizae, that is morphology, function, benefits, and isolation by two methods. First one is direct examination in roots and also examination under soil. Then we discuss about mass production of Vamp and what are the methods of applying Vamp in crops. Okay, thank you.